Sweet, umami, bitter, salty, and sour are the 5 basic taste sensations that humans perceive. Specific foods cause taste recognition of these sensations by activating various receptors in our taste buds. The concentration of table salt is also an important factor in determining taste. For example, the optimal concentration of table salt is 100 millimolar, at which point humans detect a salty taste. Higher concentrations of salt, over 500 millimolar, may be perceived as bitter and/or sour by humans, whereas very low concentrations, less than 10 millimolar, are perceived as sweet. Multiple salt detection pathways in the taste buds have been proposed by scientific studies, but their exact mechanism is still unknown. In the case of common salt, sodium chloride, salt taste sensation is primarily driven by the sodium ion, Na+. However, the anion, chloride ion Cl, is also considered to be detected through unique molecular mechanisms and participate in the taste sensation. To investigate this chloride ion detection mechanism, scientists from Okayama University in Japan have conducted a study using structural biology methods and mice models. The researchers had previously studied the structure of a taste receptor from the Japanese rice fish, Medica fish, which is similar to the human sweet taste receptor and is also structurally tractable. This fish taste receptor may bind to a chloride ion. The researchers had previously analyzed the structure of the T1R2A, T1R3LBD receptor from Medica fish, which led them to the unexpected finding of the Cl binding to T1R3LBD. They investigated whether Cl binding causes a conformational change in the receptor in this study, and were able to confirm the induction of this change. The change in structure in the T1R receptors was found to be similar to that induced by other taste substances, suggesting that Cl does activate the sweet receptors on T1R2A, T1R3LBD. Because changes in shape frequently indicate receptor activation, the researchers investigated the chloride ion activation of sweet taste receptors, the T1R2, T1R3 heterodimers, which respond to sugars. To demonstrate this, they performed electrophysiological assays on mice, demonstrating the activation of neurons involved in sweet taste signaling when small amounts of chloride were placed on the mice's tongues. Thus, they demonstrated that low concentrations of Cl could potentially produce a light, sweet taste sensation via the T1R in the taste buds. When offered a choice between dilute chloride solution and plain water, the mice recognized the taste of chloride solution and demonstrated a preference for it. Table salt is essential for maintaining homeostasis, or internal body equilibrium. The optimal intake and excretion of sodium regulates this equilibrium. The former process, according to this study, uses the counter-Cl ion to regulate the molecular functions of the receptors involved. This study's findings will pave the way for a more nuanced understanding of taste perception in organisms.